with some historic perspective. In the early 1900s, we have the rediscovery of Mendel's law of segregation by the Greeks. And this idea that two pieces of DNA can come together in a diploid individual, affect the phenotype, but then upon meiosis, to produce gametes, they can get separated again, actually solve the problem of blending inheritance that Darwin himself could answer. And so this led to the development of the modern synthesis of evolutionary biology from 1910s to roughly the 1930s. And this is also the birth of population genetics. And around the same time, Morgan's work on Drosophila chromosomes that you can see here actually put the chromosome in the center of this theory of inheritance. And there are consequences of this idea of DNA here being organized into this kind of discrete like pieces. And the consequence of that, the most immediate consequence of that we'll be looking during this talk is linkage. Linkage between sites that happen on the same chromosome. And this is because they will tend to be inherited together over generations. So jumping to the 1990s until now, we have a better idea of how levels of genetic diversity actually varies along chromosomes. And here we have the Drosophila autosomes that we saw in the previous slide. But here we have some measure of scale diversity. This was studied by Alicia and collaborators back in 2016. And we see there are some regions of the chromosome that have more diversity than others. And this is going to happen. This is going to be a general pattern for all species. We can ask ourselves why levels of diversity vary along chromosomes. So different flavors of questions can be asked here. For example, more fundamental questions, what are the evolutionary mechanisms that actually shape this variation? We can ask more practical questions. So that is like, what are the consequences of this variation? For example, diversity itself influencing phenotypic evolution over time. And we can be more pragmatic and ask what we can learn from this pattern of diversity. So we can, how can we leverage the data that we have today to make inferences about the past? So we can kind of play this game where there is some unknown evolutionary history that generates the genetic data that we have today. This is what we observe. And based on genetic data that we have today, if we understand how these processes shape diversity, we can try to guess what the evolutionary history was. For example, we can try to infer mechanisms of natural selection. We can try to infer demographic history. And we can update our evolutionary model that would then predict perhaps some slightly different genetic data and keep playing this game until we have a refined model of how evolution has led to diversity in our particular species or population that we have interested. So today we know more or less which evolutionary mechanisms are in place. We know drift, selection, mutation, recombination, migration, gene flow in general. But we are still figuring out how they interact to shape diversity, shape this landscape of diversity across the gene. Some outline of my talk. First, we're going to start with a conceptual background. Then I'm going to introduce our new model, and hopefully I'm going to be able to convince you that it works. I'm going to show then, of course, I have to do this, how other models can interpret patterns of genetic diversity and why ours is so much better. And then we're going to finally apply it to study genetic variation, and we're going to choose humans as a focal. So we resume, so some more historical perspectives in order. And we resume our journey in 1966, when Hugh and Robertson published this seminal paper on the fact of linkage and the limits of artificial selection. And this is already building heavily on the idea of a chromosome, right? So what they realize is you have, like, for example, two loci arranged in the same chromosome, they're separated by some distance, recombination distance R, and both of these loci are positively selected, then ideally, natural selection would like to fix both of these parents because they are better than the rest. But this linkage implies that they're actually going to be competing with each other. So they had a model, they had a conceptual model, and they did some simulations to test it in 1966. And this process was carried out on a high-speed computer, the LCT Atlas, back in 1963. And it was rather more abstract than that of other workers. And simulation entailed either 8 or 16 integrating. So very low population size for today's standards, of course, but they had ways to go around it. But here we see the major idea of selective interference when you have positive selective mutations is that they are trying to study the probability of fixation of one of the favorable alleles as a function of the frequency of the other favorable allele, the other loci, the other locus, and their level of recombination. So if there is like infinite recombination, so very high recombination at the top, the favorable allele has a very high chance of being fixed by lateral selection because they're effectively not inferior. And the more you drop recombination, going all the way here to zero recombination, then they start interfering more heavily. And the probability of fixation at the first locus decreases because of this, because it's competing effectively with the other locus that is being followed. So in 74, Maynard Smith and Haig took this. So this makes sense, right? So they took this to the study of link selection, again from the positive selection perspective. That is, if functional sites interfere with each other, then they must be also affecting levels of diversity at nearby neutral sites. And so this was the idea of genetic hitchhiking, when you have neutral variants that are not affecting the phenotype or fitness by themselves, but they tend to hitchhike together along with variants that are V-selected. In 1988, Hudson and Kaplan framed this process, this hitchhiking process, in Wallace terms, which is a model of ancestry among a sample of haplotypes. And in the 80s, up to 92, this is right around the time where we not only had protein sequence data, but we started having DNA sequence data. We observed, actually, for the first time, that levels of diversity tend to vary among genes, right? And so population genetics then tries to explain the observed data, this variation of diversity across genes. And this could be even like synonymous diversity, that would be relatively neutral sites. And hitchhiking is the preferred explanation. So this is the canonical selective sweep model, where a positive selective variant rises to fixation, and the nearby link variants tend to hitchhike along. So you have the emergence here in a sample of haplotypes that have surrounding diversity here shown by the difference in the presence of blue dots or their absence. So this haplotype is very run. So you have this positive selective variant that appears and will tend to, over time, eventually get fixed. And when it does so, you see that diversity and nearby link size is reduced as a result of that. So this is a process that unfolds in time, but leaves signature in space. And when I use the word space here, I mean along the chromosome. So this is like studied by Ryan Hernandez back in 2011. So about the canonical selective sweep, where we have diversity being reduced near this site, this focal site that was subject to positive selection, and it recovers around it by means of the process of recombination that tends to decouple the evolutionary history of sites in some terms. So moving into the 1990s, we have background selection sweeps through the 90s. And this is like Brian Charles would recognize that not only positively selected variants can reduce diversity, but the same thing is going to happen when you have many sites along the same chromosome that are being under negative selection. You have always to remember that today's positive selection is tomorrow's negative. And this made a lot of sense, as it makes sense to us now. So from 1994 to 1997, there was really like a series of papers that made this model of background selection more and more complicated. And so like this is collectively what we call background selection theory. So they also did simulations to test their model. The simulations, like they're doing this like roughly 30 years after Hill and Hubbardson, so their simulation is more complex. So they have 1,024 loci subject to deleterious mutations, and that population size in here is like already 1,600. So, but the idea of like having like strong rates in general was still very pervasive at that time. So we have like the mutation rates, even if you're summing all over this 1,000 rows, like, these are very large there's mutation rate. And these are super large selection coefficients, right? And so they see that the background selection model that we're gonna look a few slides ahead more in detail how it works. They see that the expectations match the observed mean of the simulations quite well, except when you have a selection coefficient of measly 1%, which is quite high. So then you don't have a good match for this particular simulation. So background selection theory entails sufficiently strong selection that the deleterious variation remains at this mutation selection balance. So you don't essentially you don't have drift in the model. So drift is not important. The population size does not matter at all in the end, according to this model of strong selection. And what you have then is this idea that the effect on leak neutral variation is well approximated by a reduction in the effective population size. So you have the rise of this idea that the effective population size is not only a property of the population, but can also be seen as a parameter that in fact varies along the and this was their model of background selection was confronted with data early on in the 1990s. Here we have some measure of DNA variation, so you can see that y-axis says DNA variation. And we have the prediction here under the background selection model. We have the observation for a few, I think like a couple dozen genes shown by this particular symbol, and their predictions according to different strength of selection showing by these other shapes. And roughly we can capture this deep in diversity that results from background selection. And the same here, we have this DNA variation as a function of the recombination. So in the end, we have like really three sides of the same coin or the same die. We have constrained variance, reduced diversity at link neutral sites, and this is going to have two flavors as we saw, the negative selection part and the positive selection part. But the constrained variants also interfere with each other because they're in leakage and tend to be inherited together when they fall on the same chromosome. So if you have like this schematic chromosome here, schematic haplotype, where the black circles represent neutral variants, and you have negative selective variants in red, positive selective variants in blue, we're going to have depression here at the black neutral sites. This is often the focus of link selection models, but these other sites are also going to be, just to summarize here. So finally, the 2000s introduced the infamous advent of large-scale sequencing that we heard so much about in grant proposals in population genetics since then, and in papers as well. But that is actually a good thing because population genetics theory was indeed developed in the dark, and it now can be contrasted with model predictions. So what are the ingredients for making this prediction? So what is, in fact, the background selection model? Well, what we're trying to do is predict diversity, pairwise diversity, for example, pi. And this is going to be a function of linkage to a series of constrained loci, M of those. The mutation rate, the deleterious mutation rate that dictates the level of the deleterious diversity in each of these constrained sites. There's selection coefficient SH against heterozygous genotype. And the recombination distance between our focal neutral sites and each of these constrained elements. When we have these ingredients together, we can multiply this production factor by the genetic diversity that would be seen in the absence of any link selection, and we have our prediction for diversity. So often we study this statistic that is the relative diversity, the relative reduction in diversity caused by link selection, which is denoted as the B value, which is pi divided by this pi naught, idealized diversity. So turns out that we have these ingredients today. We, at least for humans and some other species as well, we have for some time now the position of the exons across the genome. So we know where the constrained elements are. They're being subject to negative selection. And since 2002, at least, we have a map of the human recombination. So along a particular chromosome, we can see that there are some parts of the chromosome that recombine more than others. And this is gonna help us give or assess the genetic distance between each of our focal nucleosides that could be anywhere here and all the constrained elements that lie in the same chromosome. And so then if we assume, for example, that mutation rate is uniform across the genome, which is what this initial model did, we have all the ingredients required for making the prediction of diversity. So this is what they tried to do already in 2009 in McVicker, but they did not have high quality whole genome sequences yet in their study. And okay, so the motivation then is to study human chimp divergence, and this would be related to linked selection in the ancestral populations of human and chimps. So you see here some predictions of human chimp divergence. And I think here in gray you have the observed human chimp divergence, and in color you have the different models. So this is based on the idea that most mutations on functionally constrained sites are delivered here. So we're using the background selection model. And they use this model to make predictions. And this model offered like a reasonably good fit to the data, and it was well received by the community. This idea was well received. So jumping forward to 2022, already we have high quality sequence data. We have the 1000 Genomes Project data at our disposal. And Murphy and others were able to use the same model of background selection to predict diversity in human populations. So here we have the observed relative diversity across position of human chromosome 1 in 1 megabase windows. We have that in this grayish color. And there are predictions based on background selection theory that they seem to account position of elements and recombination map is shown by the yellow here. You see that is indeed like visually offers a good fit to the data. And indeed, this is for 1 megabase windows across the genome. But the predictive power, that is a variance in observed diversity that they're explained by this background selection model, increases with the genomic window size that you're using, the size of the bins where you're averaging statistics, and that's just because you're reducing noise, for example, coalescent noise or mutation in larger groups. Okay, soon after that, just last year, then Buffalo and Kern used a new model actually that is based on not on classic BGS exactly, but it's based on a different model developed by Santiago and Caballero since the 90s. And this model is actually very good, not only to study strong selection, but also weak selection. So here we see predictions for different mutation rates. This plot is a bit complicated, but we can walk through it. So each color is going to represent a different mutation rate. On the left here, we have stronger selection, and on the right part of the plot, we have weaker selection. The dots show simulation results, and then the lines show prediction, right? So here we see classic BGS theory as this black solid line, and it works indeed for strong selection because it makes that assumption. But then after some point here, after the strength of selection becomes weak enough, then your classic BGS theory is not going to predict well at all. But this new model can make indeed prediction for weaker selection coefficients, but it has this discontinuity problem here around NS of -1. This makes progress indeed in the weak selection region, but this is actually like the peak of linked selection you can tell, because these intermediate levels of religious diversity and intermediate levels of the pace of removal of variation combine to lead to a strong reduction factor, so lower B values, and the model does not work. So they play a game of optimizing the p-value curve and trying to fit DFE parameters and update the predicted B map and then try to guess the DFE again. So this maximum likelihood optimization model. It turns out that, for example, the DFE that they infer for coding sequencing here is highly bipolar. You either infer very strong selection or very weak selection. And this is a problem, kind of points to model non-identifiability. So to summarize previous background selection studies, so they're able to explain up to 66% of human diversity at 1 megabase scale, so they enjoy some success. But they suffer from this non-identifiability and possibly overfitting model because they're trying to learn a lot of parameters, a lot of different selective, a lot of different evolutionary processes based on the p-value curve alone. So they are unable to predict p-values around n of -1, which coincides with the peak of lane selection. They assume a constant population size across time, this N of t. And there is a recent simulation study that highlights the importance of demography actually in making p-value predictions. And they assume a constant mutation rate along the genome as well. And it's kind of because the mutation map was not available. And we know that biology is more complicated than that. Of course, we can always improve the complexity of our models. Ideally, we would like to jointly model demography, link selection, and mutation rates along the genome. So now I'm going to introduce our new model and hopefully convince you that it works. So this model is based on two locus statistics. They offer a nice compromise between the tractability of single locus summaries that may be maybe too simplistic because it assumes that size will be independent when you look at, for example, the SFR. And the more complete information contained in long haplotypes, but if like trying to model long haplotypes is also very complicated. So they offer true locus statistics, but like looking at loci in pairs offers a good trade-off between these two types of models. And we are based off this model, or the so-called Hugh-Robertson system of two locus statistics that was developed under neutrality already in 1968. And that was extended by Aaron Simon. And what they did is show how neutral processes like mutation, migration, recombination, and drift operate on these two local expectations of these two locals. And our goal here in the remainder of the talk is going to be extending their model to include selection as well. So first, some intuition about what selection should do in this two local system. So I'm going to be showing you some of these like ball and stick diagrams here. These are representing two loci. Whenever you see a closed circle, this is going to be a particular left locus that has the derived allele. Whereas if you see an empty circle, this will represent the ancestral allele. And likewise for the right locus. So we model selection against the derived allele on the left locus. So selection is going to be acting to remove any haplotype that carry the closed circle on the left. And we are going to assume that the right locus by definition is neutral for our purposes. So how selection acts, you have some sample configuration of two locus haplotypes, and any haplotype carrying the derived on the left has a chance of being eliminated. Let's say that if this guy here, this particular haplotype gets eliminated, then because we're modeling soft selection, we need to replace it with some other haplotype from the population. And you can see here that by having this process of selection eliminating preferentially two types of haplotypes, this is going to change the statistics over time. So we work on expectation, and we say that in the end, our model works such that selection steadily removes a fraction of haplotypes carrying the other two results. So S is going to be negative. And oops, here. Okay. Making it a little bit more formal. I know this is it's going to get complicated, but we can walk through this figure. So this is our two locus model. We have the right locus being neutral, so S is 0. The left locus is being under some strength of selection, which we call S. They are linked by some recombination distance R. So you can imagine that the closer they are together in the chromosome, the lower the recombination rate is between them, the more the right locus is going to be affected by link selection. So we have a few statistics here that we're trying to model. Phi L here at the left is genetic diversity at the left locus. And this is going to be affected by, well, the demography, denoted by N of T. some mutation rate that is dictating the rate of input of these deleterious allele on the left, and some selection coefficient that is removing this variation. On the right, this guy is neutral, so he's not affected by S directly. He's affected by N of T and U, of course, but he's affected due to linkage or LD, linkage disequilibrium, together with the left one. This LD here is also affected by, for example, N of T, mu, S, and also the recombination. Okay, so this is a pure two-locus model where our focal neutral site is linked to a single site that is under selection. How do we extend this to, well, let's say many constraint elements along the same chromosome? We do the same thing as Hudson and Kaplan did. Then we say that to predict diversity at a focal neutral site X by R for the right locus at X, this is going to be a function of this idealized pi naught level of diversity at a focal neutral site X, and then we're going to multiply together this B value, this relative reduction coming from the interaction of our focal neutral site and all of this surrounding constraint sets. So the B value or the reduction that X experiences from a constraint element that is close to it is going to be different, of course, is going to be stronger than the link selection effect that it experiences from a constraint element that is far from it, right? Makes sense? Because the recommendation is 0. In the end, we're going to multiply all of this together to get a prediction of X, of pi R of X, that is a function of all the relevant, all the constraint elements that should affect it. So in the end, we can put this all together in a system of linear equations. And we can represent this by matrix operation. So we have here, we have the D represents a drift matrix that represents essentially the demographic history of our evolutionary model. We have some matrix U that represent how mutation affects these two local statistics. A matrix R that represents how recombination affects them. And finally, the selecting matrix that we derived here. When we multiply this together, we end up with a transition matrix M that represents all the relevant evolutionary processes that are affecting our two local statistics. And this transition matrix is going to be affecting forward in time the evolution of a vector of summary statistics here, that where you have some LD statistics, some two local statistics, a collection of them. We are not going to be looking at them in detail, but they're required to perform this prediction. We have pi L, diversity at the left locus, and finally we have pi R diversity at the right locus. And the index here, I, represents the particular epoch that we're looking backwards in time. So when we have the demographic history, there is not constant. For example, we can look at this 3-epoch demography here. We start at the top at our ancestral population that has some size NA. And we start a steady state for the summary statistics. That means that we are representing diversity here in this ancestral population by this factor V. To get diversity or these two local summary statistics here at this particular point in time, what we do is multiply the previous vector from this previous epoch by the transition matrix that has the parameters of this epoch, and this is going to be applied for T1 generations, where T1 is the amount of time in duration that we spend with this new population size in 1. And this is going to give us the prediction at this point in time. To get prediction at present time, let's say we sample present day individuals and measure diversity in them. Here at the bottom, then we take this vector from V1, so at this particular point in time, and apply the matrix M2 that refers to this new updated population size. And we apply it for T2 generations, which is the amount of time that we spend. Finally, we have the predictions of our summary statistics here at present time when we sample our individuals, and we can look. We're going to pay particular attention to this priority. So we can benchmark our model first under equilibrium, that is assuming that the population size has a degree remaining constant over time. we are plotting for different mutation rates again in colors, this diversity reduction in p-value as a function of selection coefficient. So here we have weaker selection, here we have stronger selection. We have the simulated dots, the simulated values shown in dots. And we have different types of prediction. So first, we have the classic BGS prediction, and as we saw before in the other slide, the BGS does a very good job when selection is strong, but after some point, no longer. And then we have prediction using our model. We have the naive prediction that is shown by this solid line. And we have another type of prediction that accounts for interference among the constraint sites in the same chromosome. And that is shown by the dashed line. I'm not going to get into detail how interference works. If you don't, you can ask me later. But the important part here is that it does indeed improve the prediction a lot in some cases. And this is simulation of a 100 kb segment of the genome that is fully constrained. So we expect interference to be quite high in this particular scenario. We're just showing here the interference that we're acting for. It does indeed do a good job of recovering the simulated values. Then you see here that there is no discontinuity in the prediction around an S of -1, which is important. So when we simulate a more complicated scenario, here, for example, the first 30 megabases of human chromosome 2, this is the recombination rate that we use for different parts of the genome of this particular chromosome. This is coming from an empirical recombination map. And we use the position of exons as present in human genome 38. We see that our predictions here, shown by color, agree very well with the simulated data forward in time, which is represented by the blue dots. And there are some observations that we can take from this particular plot right away. Indeed, when we have exons, diversity tends to reduce in these particular regions because they're traveling together with more constrained elements. They're being subject to stronger link selection effect. And this is going to be true in particular when the recombination rate in that region is low. So we can contrast this part. There is a very strong reduction in V values relative to this part. They both are exon rich, so to speak, but this has a higher recombination rate than this region. And in the end, the diversity drops more. So, on to the novelty, the conceptual novelty of the model. We can now model B-value evolution over time. And has already been shown by simulations that there can be a strong interaction between demography and selection that leads to non-monotonic behavior. So here I'm showing you the B-value. Now this is no longer the position of the chromosome, but it represents generations ago. So this is coming from a pure to locus model, just to show you the behavior that we would expect. We are testing two demographic scenarios. On the left, we have a tenfold bottleneck here, so a population that starts from size 10,000, and size population size 10,000 that suddenly drops to 1,000. Or the same population of ancestor size 10,000 can suddenly increase to 100,000. Here I'm showing you for selection strength of an S of -10 to -3, which in the end with this population size represents fairly strong selection. So starting in the ancestor population time is presented in generations ago. So we start from the right and move towards the present, going to the left in each of these subplots. Ancestor population going to the present time. We see that the p-value, depending on, like for all recombination rates, so we have three different recombination rates here presented. And the recombination rate does not change the shape of the curve itself, it just changes quantitatively how the p-value is. So first, after population size change, we have this sudden drop in p-value in the bottleneck scenario. And then we have this recovery that goes beyond the previous equilibrium point. And we have this recovery that goes beyond until we find a new equilibrium and we stay the same. For the expansion scenario, for the time considered here, that is 120,000 generations, we have this shape here where you increase B value and you don't find the equilibrium still. Because an expansion scenario is limited by, the new equilibrium is going to be limited by the rate of input of new mutations, which tends to be slow. And so you can take my word that I did this for a longer time, and after 2 million generations, these values eventually drop below the previous equilibrium point. So again, showing non-monotonic behavior. So finally, we move on to trying to show you how previous models can misinterpret these patterns of genetic diversity in light of the importance of the demographic history for the prediction of B values. So in other words, what are the practical consequences of neglecting demography? It is reasonable to think that biases in the prediction of B values should carry over to downstream inputs, like those of the DFE and the diffuse mutation rate as performed by Murphy and others, as well as Buffalo and Kern. So we investigate this effect in a very simplistic scenario where we have a simple chromosome layout with uniform rates, that is, recombination rate is the same and mutation rate is the same all across this chromosome. And the position of the constrained elements is as well evenly distributed. And we project the system forward in time using our bottleneck and expansion scenarios. And our approach is to predict equilibrium B maps at different points in time and contrast it with the demography aware B map using our full model. And by doing that, we can perform inference, assuming equilibrium, and try to recover the simulated DFE, or distribution of fitness effect. And this is going to be a DFE that follows a gamma distribution that has two parameters, a shape and a scale. And so this is like a distribution of selection coefficient. I'm showing you here like 3 example DFEs just to so you guys have an idea of what this looks like. So we have some probability density here of being in a particular of being a particular value of X, a particular strength of selection. And so the gamma distribution is supposed to be quite flexible. So the scale should tell you more or less where you're standing here along the x-axis, and then the shape is gonna tell you how spread your distribution is for this particular selection coefficient. So we can take on very different shapes depending on your parameters. So here we are trying to infer the shape and scale parameters of the DFE, or the relative mutation rates, that is the deleterious mutation rates influencing deleterious genetic diversity as our constrained elements. Under our 2 scenarios, in shades of blue, we have the bottleneck scenario, in shades of red, we have our expansion history. Again, time flows from the right to the left on each of these subplots. And what we see here is that if we take for granted and we assume the true mutation rate and try to infer the shape and scale, when we start off in the ancestral population, then it's nice, we can recover the 2 parameters because equilibrium actually holds in the ancestral population. After the size change, however, the more we move into the bottleneck population, the bias in inferring the shape by assuming equilibrium grows at first and then shrinks back when the new population size becomes the new equilibrium population size and the assumptions of equilibrium are holding. However, as we saw before, the expansion history does not find the new equilibrium value within these 25,000 generations that we tested. So the bias grows indefinitely in the time period that we consider. Likewise, for the scale parameter of the DFE, here we have the true values shown by these dashed lines. And for all of these, bottleneck, the bias grows and then shrinks as the new population size becomes a new equilibrium. And the bias in the inference of the scale for the expansion history goes throughout this 25,000 generations. When we take the DFE for granted and we fix these two parameter values here shown in parentheses, and we try to infer this time the deleterious mutation rate, and we have the relative mutation rate shown by the dashed line. So one means that we are recovering the true value, right? So starting from the ancestral population, nice, we can recover the true value. Going into the demographic history, the bias grows for the bottleneck and then shrinks, and grows indefinitely for the expansion history. And eventually, if we wait 2 million generations, then indeed we will be able to make it. This is going to go back eventually to the true parameters when the new population size, this 100,000 population size becomes the new equilibrium value. So finally, we're going to apply our model to study genetic variation in humans, and this is done. This is work done together with my collaborator Nick Collier, that is also a member of the lab.
You have about 3 minutes, just so you know.
How many?
3 minutes.
3 minutes.
Yeah.
Right. So what should the human landscape of diversity? You can ask this question. We have some observed diversity level here, and like we have for Drosophila in the introduction. So we can ask, yeah, what is dictating that? We can start now that we have the roulette mutation map, which was recently published by Stiplewski and others at the end of 2023. This is what the roulette maps look like. So this is going to tell you, it's going to tell us deleterious diversity, sorry, the input of deleterious diversity in different parts of the genome, but also the input of nutrigenic variation in different parts of the genome. Given that we have this roulette mutation map, and also using the updated human recombination map inferred by Jeffrey Spence and others in 2019, and knowing that we have either I have like 2 minutes.
Yeah.
Let's see. What is the approach here? There are different ways to classify the constrained sites along the human genome. We can use a functional model that is going to take into consideration the position of exons and also regulatory elements like promoters and enhancers, or we can have fast models where we identify sites under constraint using a phylogenetic conservation score in a multi-species alignment. And we are going to use for all of these classes of constraint elements, we're going to use the DFE. We're not going to infer the DFE ourselves, but we're going to take it from elsewhere. So we're going to get those DFEs, distribution of fitness effects and selection coefficients, from methods that are based on the site frequency spectrum, which are independent from our model. So combining all these elements, we can have a prediction of diversity for our two classes of constraint elements. And in black, we have the observed diversity, and then in orange, we have the model based on the lean selection model based on fast funds, and in blue, we have the lean selection model based on these functional elements, exons and regulatory. And you see that they offer a very good fit to the data. This is in 1 megabase windows for human chromosome 11. In general, the fast funds models tend to do better. And in the end, this is showing you chromosome 22 using 100 kb window sizes. And you see that it becomes, of course, more jagged here, but the model does a very good job of predicting diversity and particular state still for chromosome 22. And in the end, this is not so different from those original predictions from Hassan and Kaplan back in 95, except that now we have a more complete model of evolution in human chromosomes. We can ask how well does our model explain these diversity levels? We can assess that in a linear model framework by asking the R squared, so how much variance in diversity is predicted by the model as a function of the window size. And the variance grows with the window size, as it did for other studies as well. And we see here for chromosome 10 1 megabase window, 1 megabase window size. Then for fast-ones model, we can predict just above 60% of the variation. And likewise for chromosome 21, we are comparing here either the exons regulatory or the fast-ones model. They are quite similar. And we also, when we plot this, Here we have the total R square for different chromosomes. And here we have the total R square of the model for different chromosomes according to window size for the fast-month model. We see that there is a large degree of heterogeneity in the variance that can be explained by the link selection model as a function of which chromosome we're looking at. This is interesting itself. Unfortunately, I cannot get into detail right now. And so finally, to summarize, here I've shown you like a new model of link selection that works under non-equilibrium homography and the full distribution of fitness effects that is under a wide range of selection coefficients. Inference from previous studies were biased due to overfitting and model non-identifiability. Even without playing the game of maximum equilibriumization, our R square is on par with the previous studies. And furthermore, we found this large heterogeneity in R squared among chromosomes that was previously undocumented. So there are caveats, the roulette map, that could be a bias in the interest of the roulette map itself, mutation map that we are still investigating. Simultaneously accounting for demography interference is very challenging, and this is something that we can discuss later as well. We are currently restricted to single population models, and we can imagine that if non-equilibrium demography, there is varying population sizes for a particular species, a particular transmission species, already leads to complex B-value dynamics, then if we have two populations that have some degree of structure and gene flow, that can also lead to some very rich patterns that we hope to explore in the future. Thank you.
So sorry, we do have to be out of this room now, so we won't be doing a public Q&A. Thank you, everyone.
Class could move to 1316. I suppose if anybody from the public has any burning questions, you can come to 1316 and we'll let you ask them first.